My name is Sarah Owens. I'm a baker, author, and horticulturist, and I am leading the sourdough track, which is basically new frontiers in sourdough microbiology. With the 20 students that we're leading through this track, we're basically looking at 6 different sourdough cultures from all over the world, including Alaska and Belgium and New York and California. And we're studying the differences between their aromas, their performance, and sort of evaluating them in one particular dough formula. So this particular track is really focusing on enlightening ourselves as bakers in terms of anecdotal evidence of things that we've observed over hundreds of years and bringing it into the present and also trying to pull it into the future of bread making. Mostly by making observations and then analyzing that from a microbial perspective. And a lot of this sort of technology that we have available to us now through different lab techniques of isolating strains of yeast or bacteria are enabling us to have this conversation in a really particular way that we've never been able to do before. And the symposium is really about sort of the future of bread. And so On the Rise, the title of the symposium this year, is really, it's all about understanding how do we get that rise, how do we get that flavor, how do we get that crust or that crumb in our bread, not just from a baker's perspective, but from a microbial perspective as well. And so what we found is that sometimes these cultures being fed different types of flours, they're in a different type of ambient temperature, they're being handled by different people, they tend to express themselves in a really different way. And so we're finding some surprising results. We're also finding that some of our assumptions were true in terms of flavor, rise, fermentation activity, and baking performance. So it's been kind of a mixed pan of results, but what's interesting is that we can have this conversation about, well, why would that be? You know, let's really look deep into what actually happened and how that would influence the microbial sort of ecology of each of these starters in our dough, and of course our final bread.